it's kind of like learned helplessness. So our gals in the office, we have 4 T's. Can you believe it? Tara, Teresa, Tracy, and Trudy. And those are our 4 gals in the office. They're all good. And it's like learned helplessness because they're so good. I don't know how to do squat anymore.
Huh. Yeah, that's easy to have happen.
Well, it's a nice position to be in. Not all the departments around here are even close to that. I'm sorry to say, but not us. We got great people, which is great for us because yeah, this is just parts of my brain that I don't have to keep going. Yeah, but at any rate, that's why I was great. The title was perfect. So this is the kind of stuff that all these marine people like, hey, that guy's using words that we use. He's using data sets that we have. What's that all about? So I said, yeah, find out how the Eastern Oregon desert, high desert, is linked to the oceans there. So
yeah,
I already had some of my colleagues say, are you just pulling our leg? And I said, no, I'm not. I said, you got to come to this one. Yeah, it'll be good. This will be good. It'll be nice. Whoop, they're starting to trickle in now. Let me see attendees. Oh, this one, I have all the power. When you're in this thing, you can't see all this stuff.
No, I can't. And that's
you probably can't even. Oh no, you could see it. Yeah. Oh, Sylvia's there. Hi, Sylvia. All right.
I can see you, and I can see a beagle.
That beautiful beagle. That's Teresa's girl there. That's one of those little miniature beagle, not miniature, but the tiny bit. She's something else. She's a heartbreaker, that one. Get us in small here so I can still read stuff. Whoops, get out of there. They'll start coming in. Where's my clock on this thing? I better get this one out. I might start a minute late. All right, we're at the top of the hour, but like I said, I think they'll squeeze in. I'll give them 1 minute. I'll be 1 minute late here. Oh, that's our 1 minute right there. All right, let's see. Hopefully everyone can hear me. I think I'm talking to everybody. So welcome to the department seminar, and it's a real pleasure for me to introduce today's speaker, Pete Zaney, who I've known, I think, for as long as he's been since his graduate school days, because he went to that other large institution to the south of us, about 45 miles south of us, where he got his PhD, and he worked with Amelia Martins, who some of you might know. I think she's still at IU, but maybe she's moved again.
Arizona State.
Arizona State. That's what I was going to say. I thought she had. Yeah. So she had moved to IU, Indiana U, and now she's at Arizona State University. But at any rate, someone's done a lot of work on social behavior in lizards and so forth. And that's what Pete has worked on. And so he has a very, I think it's a great talk for this group and, our campus in a broader sense, because he is really done some excruciatingly painful, not painful, but very difficult field studies. Like every data point in this talk is something that really took a lot of effort here, trying to catch these little lizards out in the Eastern Oregon high desert is no mean feat. And so, and then Pete has been doing this for years now, year after year after year, compiling what I think is really a unique data set, especially with an untimely loss of one of our colleagues down to the south of us in California. But at any rate, and so when you see his title here, you'll think, whoa, what is this guy talking about? But I think this is a talk well worth attending to look and see how these small lizards at the, way out in our Eastern Oregon desert are severely impacted and following things like climate conditions in the oceans and not weather conditions, but climate conditions. And so anyway, Pete's going to tell us all about this. So let me not steal his thunder anymore. I should say at the end, in case we go for you grad students or whatever that might be reticent to ask questions, Pete would be happy to interact with anybody after not right after the talk, but just send him an email. I can hook you up if you can't find him. He'd be happy to talk to anybody about this stuff. And so what we'll do is what we've done in the past is, just because it's this kind of weird Zoom webinar thing, is just put your questions in the chat, not the chat, in the question and answer section, and then I can either read it, or what I'm going to try to do is turn the microphone on so you can ask your own questions, then you can follow up. All right, so without further ado, Pete Zaney is going to speak about influence of Pacific Ocean climate modes on the life history of lizards in Oregon's high desert. ENSO and PDO affect clutch, timing, and number. Pete, go for it.
Thank you, Bob. I really appreciate the invitation. It was a great opportunity for me to understand my own data better. And I just want to start by saying, 20 years ago when I was finishing my PhD at the University of Oregon, I was struggling to figure out what to do with the rest of my life. As many graduate students do, trying to complete the degree becomes so consuming that it's hard to know what the next step is. And as someone pointed out to me at the time, whatever it is, it should be important. It should matter. It should be something that makes a difference. And so that's what I tried to do then, was to establish an understanding of the life history, something that I'm interested in, of lizards in Oregon's high desert, to try to understand how climate and climate change, which at the time was becoming apparent would be the overarching influence on populations and species moving forward. And trying to do so then, I'm trying to understand how the El Nino Southern Oscillation and the Pacific Decatur Oscillation, both of which have teleconnections with weather and influence on events in Western North America, how those climate modes, those natural oscillations in the Pacific Ocean, impact a terrestrial system like what's going on in the high desert. So achieving that broad and general applicability has really been my goal over the last 20 years. And in order to do so, what I've tried to do then is understand what's going on with these lizards, the common side blush lizards. And this is a typical population out in Eastern Oregon. This is the north end of Lake Hebert. And as you see, they live in areas like boulder fields, along scrub, the desert, the sage steppe environment throughout the Pacific Northwest, but also in many other areas, as I'll get to. Before I do, I want to give you a little bit of background as to what leads me then into the main focus of this talk. If you read the news, what you realize very quickly is that there are almost daily reports of things like species declines or extinctions that are going on in plants such as trees, or in marine systems like the tuna, or in freshwater species and terrestrial species, that the news is universally dismal in many ways. And if you look at the literature, it's climate change that appears to be one of the major drivers of what's going on with species extinctions, with local extirpations of species, collapses of ecosystems, that we see example after example in the literature focusing on climate change as being this overarching theme influencing populations. And that then really is my goal, is to understand how climate change is impacting populations of lizards, using the Pacific Northwest as an example, because it was relatively underrepresented in studies of these kinds of organisms. So how is climate change affecting individuals in populations? How is it coming together to affect populations and their persistence? How then are species able to persist in the face of ongoing climate change, assemblages of species and communities? And then how is that going to impact ecosystems? My own focus has been on life historical changes related to things like reproductive timing and effort, because of their impacts and potential impacts on populations, and the potential for those populations to either benefit or be harmed by changes to those natural systems. What I also want to do is focus on something that's simple, because simple metrics are going to have a potential to last longer and have a longer broad-term effect than something that's complicated or hard to replicate. So changes in body size distribution as a simple illustration of something like population demography and the age structure of a population and its role in the persistence of those populations. And as part of this, I think it's important to think about what our data legacy will be for future generations of biologists, because future understanding of ongoing climate change beyond our lifetimes will become critical. If we start with the assumption that the things that we're doing right now matter, we need to ask ourselves, how will future generations of biologists be able to answer those how questions? And that's only true if we can give them a relevant data legacy, meaning the empirical data that we provide today as a before to the after climate change has occurred in 50 years, in 100 years, in 150 years and beyond becomes a really important question. But it's the question that's especially hard to address because it requires that we predict which species will go extinct and which species will remain extant. And those extant species then become very important because if we don't provide the data legacy that's relevant, then we're limiting the understanding beyond our own lifetimes for those future generations. My candidate for a species that should be able to persist long-term are common side blotch lizards. Common being the key word in their common name. These are small lizards, only about 40 millimeters, so 2.5 inches body size at maturation. They get up to about 55 millimeters for the largest individuals in populations. They're really abundant where you find them. In the south, in Mexico, maybe 10 individuals per hectare, but in coast range of California, several hundred per hectare. In Oregon, typically 75 individuals per hectare isn't uncommon. This picture was taken in Nevada, about an hour east of Reno, right along the interstate. And you can see the boulder field in the background that these lizards live on. This is along the shores of ancient Lake Lahontan. And the lizards that live there are living in this really desolate environment from our perspective, but at about 50 or 60 animals per hectare. And they are the classic R-selected species, the live fast, die young lifestyle. These are early to maturation, early to mature, heavy breeders with lots of loss because of predation. They're the basis for a lot of higher level predators, such as other lizards that prey on them heavily, snakes, birds. They have a really broad geographic range as well. They span nearly 25 degrees latitude from the southern tip of Baja California up to Washington State, and from the coast of California at least over to Oklahoma and Texas. And so they have a really broad geographic variation that shows up in terms of the phenotypic variation present in the species. On the bottom right there, you see a male and a female of Euta stansberriana stasnegerii in Texas, a very stripy subspecies with lots of color variation, different from other populations, such as what you see in the bottom left there, Euta stansberriana elegans in Nevada and California. More chevrons in these populations, different colors show up with lots of color variation. So again, lots of phenotypic variation in this particular species. On the Colorado Plateau in the upper right, you see Euta stansberriana uniformis, where the males and females can be really hard to tell apart because of the lack of secondary sexual characteristics in terms of coloration. In Oregon, Euta stansberriana nevadensis, the upper left, you see the lack of pattern in the females, but the males have that really bright orange lateral coloration, most likely because of color matching with the local lichens, although that's never been demonstrated in the literature. So lots of phenotypic variation, which probably relates to standing genetic variation, which is going to enable populations to adapt to ongoing climate change. Here's a map of the elevation of the Western North America color-coded. I've outlined the 5 different subspecies that are presently described. The map doesn't just stop in Mexico. They go all the way down to the tip of Baja, extend down the coast, down into the Chihuahuan Desert as well, over in the east. You can see where they occur in the Bonneville Basin, up on the Colorado Plateau in pink there. And something I'm not going to have a chance to talk about are some of the interesting work that I'm doing related to the hybrid zone that's occurring here in the Grand Canyon region. And what that picture at the beginning showed, was a backpacking trip this past summer, about a 900-meter elevation difference from the Roaring Spring, where they occur up on the North Rim, all the way down to the river. And we collected animals both sides of the Colorado River at different elevations, replicate samples on both sides. And they're really interesting because they're super common in the Grand Canyon. It was super easy to collect these things. They were everywhere we looked. We didn't have any problem collecting them. What you see in the bottom right is an animal that's active right along the Colorado River. You can see Silver Bridge there in the background, and you can actually see that the sun has set down in the canyon bottom. This is late in the day. This is about an hour after the sun has gone over the rim. That's when the animals come out. This was a 120-degree Fahrenheit day, so a super warm day. Animals are inactive in that cobble bar. That's the picture from the bridge of the site. It's about a football field size cobble bar right along the river. But the animals are really abundant with other lizards present as well, right down to the water's edge. So they're highly adaptable in the Colorado River environment. Everywhere you find water and water subsidy, then you find the lizards. And what we were doing was collecting DNA for the hybrid zone study that I'm taking part in. What this is showing is a zoom in of the Grand Canyon itself. You can see there in the bottom left the elevational difference of the Grand Canyon. In white are dorsal scale counts that I've collected as part of this study, and in yellow are dorsal scale counts that are present in the literature. And this is a trait that's been reported in numerous studies. It's a good subspecific identifier. And what this is between the parietal and the back of the head down to the posterior margin of the hind limbs. If you simply count how many scale rows there are, you can tell that up on the Colorado Plateau, in the Page vicinity, there are over 100 scale rows per individual. And that's a pretty consistent trait across the Colorado Plateau. And then if you go down the Mojave, over by St. George, and then down from there, you actually get fewer than 90 typically, down in the 80s for the most part. And what I did was I collected populations on either side of the Kaibab Plateau. And what you can see is on either side of the plateau, there's actually a gradient consistent with the hybrid zone that McKinney described in the 1970s, and show that there are differences across that gradient, that 100-mile or so gradient that seems to be the hybrid zone between Page and St. George, where the dorsal scale rows gradually decrease across there, with the Kaibab Plateau really sitting as a genetic barrier, as a high elevation barrier between there. The Grand Canyon samples that we collected on either side of the river are significantly different, so that we get more scales on the south side of the river than on the north side of the river. So it seems like what's happening is the river itself is acting as a barrier. And then the elevational gradient that's created by the transition from the Mojave up to the Colorado Plateau is a second gradient. And you can see this repeated in the samples that we collected when I put on there the scale rows for each of the 7 sites, that higher up, you typically get more scale rows than lower down. So there's potentially 2 different gradients going on across the river and along the river gradient that are creating some really interesting hybrid zone dynamics that are ripe for investigation. And my future plan is to sample along the river to be able to get a sense of where and how that hybrid zone is established, and whether or not it's a permanent zone or if it's a moving zone between the 2 different environmental conditions. What this does, though, is it brings me to my next point, which is across its range, side-blotch lizards are really a riparian lizard. Along the river, found them in many places along those cobble bars, but also along places like Garden Creek, at different places, different elevations, wherever you find that water input. It's really the water subsidy of insects that allows these lizards to persist. However, in many places in the Southwest, they're considered a wash lizard. And that's because they can persist in areas where you get that seasonal pulse of resource from the water flow. So this is just below Burro Mesa Pour-Off in Big Bend National Park, showing Javelina Wash. And those dry washes, you can see Santa Elena Canyon in the background, are full of side-blotch lizards as well. So it doesn't have to be a flowing system. And like I showed you before, along ancient Lake Lahontan, the ancient lake shores become extremely good environments to look for them. That's true in Oregon as well. Summer Lake, Lake Bear, those ancient lake shores, because of the rock habitat, become very good habitats for these lizards to occur. In wetland environments, you get the lizards that occur in Oregon in particular, in rim rock that are adjacent to wetland environments, such as occurs in the Malheur Basin. So the Malheur wetlands, which are off to the right in this picture, seasonally flood from the mountains that surround it, and the lizards live in the sage steppe environment surrounding it. And because of that input of insects, they are really abundant in this particular environment. Here's a picture that I took off the Harney County Sheriff's Facebook page, showing you the field site out in Eastern Oregon. And it's surrounded on 3 sides by wetland. And that seasonal input from the Blue Mountains to the north, as the water then transitions down to the Malheur Lake off to your left, provides that input of resource that the lizards really rely on and really benefit from. So even a year like this, where it was really wet, or the current year it was especially dry, the lizards don't seem to be limited in the same way other populations are, because the resources is in essence providing unlimited food for the lizards. You can see the background picture of the upper right showing you the habitat sage steppe environment where these things live. This is a picture of the ventral surface of an adult female lizard that's gravid in this case. And it points out something really important, which is, unlike other populations where the animals don't really get this old, the population that I'm studying has such age structure that the females really have a chance to age into some interesting phenological or phenotypical traits, one of which is this nuptial coloration on the venter. That blush, that orange blush, only shows up when the lizards first ovulate, and it persists as long as the breeding season lasts and slowly fades over the course of the summer. So that you can pick up an animal in the spring and tell where it is in its gravidity cycle, in part based on that orange blush, but then you can also feel between your fingers in the abdominal region the follicles and palpate the animals and tell the gravidity, the phylogenic state of the animals themselves. The other thing about this population that's really nice is things like collared lizards and leopard lizards are distributionally limited to the other side of the steens and don't occur at this site. There's a few snakes, rattlesnakes, racers, and gopher snakes, as you might imagine. But generally speaking, birds are probably the main predator, and the other predators simply aren't that abundant at this site. So we get age structure in this population. I cut off at 3 years old, but the marker capture studies that I've done over the past 17 years, I regularly find 7-year-old animals at this site. 7-year-old animals are more or less the same as a 3-year-old animal in that they've reached asymptotic body size, which is what you see here in the female and the 3-year-old category. 2-year-olds are slightly smaller, 1-year-olds are slightly smaller. Early in the season, you can tell the early season from the previous year, young of the year, and the late season clutches from the previous year, young of the year, both of which by the end of the breeding season will have reproduced, matured and reproduced within that first year. So what that means is the older females enter the season ready to go. They've, as that good ectotherm strategy uses, stored energy from the previous summer, have that stored energy ready for yolk deposition so that their first clutch is really a capital breeding strategy. And then the second, third, and subsequent clutches are income breeding from whatever resources are available. 2-year-olds take a little bit longer. We know from previous studies I've done in my lab that there's a temporal component to the reproduction, that older females reproduce first and the smallest, the youngest females reproduce last because they have to mature in that first breeding season. So what we can tell then is the phylogenic cycle from palpations on a 0 to 5 point scale. And truly, that's a 5-point scale because 0, lacking follicles, is the same as post-reproductive, except that the animals have that orange nuptial coloration as an indicator that they've completed their first clutch. And then by feel, with practice, you can tell unenlarged follicles as these small enlarged things, BB size enlarged follicles. They get soft when they yolk, and then they get oblong and firm up when they get shelled. And so that's what I'm going to post on the plot on each of the y-axes here, gravidity scores. And it's always going to go from 0 to 5 in every case. And in each of these plots that I'm going to show you, we're going to start at the top in late April or early May, so early in the season, and progress down through time, down to about mid-July. And I've consistently done this from mid-May to mid-July over the past 17 years. And those are the data I'm going to show you. This is just the first of many years that I will talk about. On the x-axis is the body size with the age categories and shaded. So the 3-year-olds, 2-year-olds, and then all the 1-year-olds below that. And what matters really isn't the body size here as much as the relative age categories that in 2011, the most limiting year that we saw because of the La Nina that happened that year, the weather was so harsh for the lizards that they really didn't start reproducing until early June. That's when the first clutch was produced, was June 7th that particular year. So that when you go out there in mid-May, you just start to see the biggest females at enlarged follicle stage. And the smallest females haven't really started that yet. By mid-June, you see animals that are entering category 5, meaning they're post-reproductive. They've laid their eggs, but they have the orange blush. You can tell it's their first clutch. They're not lacking follicles. They're after the first clutch. And then we see a progression in time, that nice slant showing us that there is a size component to this, where the youngest females really are the last to mature and enter the reproductive stages. It becomes more challenging then when we see the bottom category, that last panel, where in early July, it looks like it's a hodgepodge. And what's really going on is different clutches are being produced so that we see the first clutches show us this nice progression through time. And that will be the consistent thing. Down through time, you'll see the clutches move from right on this figure to the left. And I've color coded them so we go from green, the first clutches, to yellow, to more red colors for the third and subsequent clutches. So 2011 is the nice year to show as the first year because it's the simplest year. It's the year when the biggest females got 2 clutches, the smallest females got 1 clutch if they were lucky, and there really wasn't more reproduction than that. 2021 as a counterpoint, was in early May already with females who were in the largest age categories as post-reproductive, meaning they'd already laid their first eggs by the time I got there. And that's part of the challenge on an academic schedule, to get there early enough to see this reproductive strategy that's going on in real time. So that by late May, those animals are starting the largest ones at least, starting their second clutch, whereas the earliest ones are just finishing up their first clutch. The smallest ones, I should say, are just finishing up their first clutch. We then see a potential for a third clutch, and then in this case, I scored a fourth clutch for the largest size category, those 3-year-olds or above. And in case you're wondering, the potential is for them to get 7 clutches. That's what some research back in the 1970s demonstrated, that theoretically these animals could do up to 7 clutches in southern Nevada with enough of a breeding season. Empirically, they saw in that particular study 5 clutches. So they weren't able to empirically show they did 7 clutches, to theoretically show with the breeding season that this population in Oregon has room to grow, that 1 or 2 clutches is a potential catastrophe from the point of view of the population. And what I'll point out is these lizards really do need 3 or 4 clutches for the population to be at this large, stable size over time. So this is all 17 years worth of data that I'm showing from the upper left, 2005, across the top going 2005 over 2010. And then 2011, this is the year I just talked about, showing us the most simple version of this, where the climate, the La Nina that particular year, delayed reproduction to the point where they only got those 2 clutches. There could have potentially been 4 clutches in some years like 2012, 2014 or 15, but because of the collapse of the population after 2011, with that poor showing in the breeding season, what happened is the age classes didn't exist in 2015, for example, for the animals to really reproduce. What we see is there aren't those 3-year-olds in the population anymore. So that even though they might have gotten 4 clutches if they were around, the lack of proper age structure in the population precluded the population from really responding to that permissible environment. 5 of the last 6 years have had 4th clutches based on this scoring. And that 4th clutch really becomes the thing that allows those individuals to expand the population. And I should point out, this is based on over 3,000 individual palpations of females over the course of each of these springs. And again, we're going from early in the season to late in the season in each year, with the color coding being the thing that really matters, getting down to that 4th clutch. The clutch iterations now is in part driven by the environmental variation that we see going on out in the Pacific Ocean, as I'll point out in a second. These are all animals with intact tails. Tails that are at least 50 millimeters or longer is what I consider intact. Adult female will have a tail that's somewhere between 65 and 70 millimeters long. Why intact tails only? Because if you have a large regeneration or large loss of the tail, the energy loss potentially limits your response energetically. You have to replace that energy. And so now what happens is it could increase variation in the data that we see here. So this was just to make it cleaner, to be able to see with just those 3,000 animals with those intact tails. And I'll show you additional data in a second that includes animals with all tail regeneration types. So this is just showing you that there's variation from year to year in the number of iterations they get. But it turns out that the Pacific Decadal Oscillation and the El Nino Southern Oscillation really don't determine this so much as the local weather conditions in that particular year. It just so happened that 2011 was a La Nina year that drove that weather change. But so far, with only 17 years worth of data, I can't definitively say that either of those oscillations out in the Pacific are what's causing the iterations to change. So what I wanted to do was focus on a simple demographic trait to understand what's going on in the population as a dependent variable. And what I've chosen is to understand or try to understand what's going on with respect to the body size distribution. The problem with that is if we only include animals with intact tails, what's shown in the red outlines here, the body size of those animals will mean that older animals have a greater chance of losing their tail, so that there's more animals that aren't under the distribution that is intact tails. So the black is showing you all the animals in 2021. The red is showing you only the intact tails. So as they age, they're more likely to show up as regenerated tails. And so what that means is it potentially includes a bias that is age related if we only include intact tails. So instead, I'm going to focus on all the animals in the population in this case. Here's 2021 showing you the different peaks correlated with the age structure of the population. And I said before, I really categorize it as 3-year-olds, 2-year-olds, and 1-year-olds. And it doesn't quite line up with the body size cutoffs that I use of 51.5 millimeters for 3-year-olds or older. 2-year-olds is between 51 and a half down to about 47 and a half, and then under that being a 1-year-old, but it's pretty close. The problem with this, as well as the clutch iterations, is there isn't any one value that really incorporates all of the variation in that population. The average of each of those age classes isn't something that we can easily see the variation of. So instead, what I'm going to do is focus on the average age or average snout vent length of the whole population. A snout vent length is simply the tip of the nose down to the cloaca, the standard body length and reptiles measurement. So if we use that average, the question then becomes, how is the average changing over time? A recent study that I published in 2018 demonstrated that the average shifts based on conditions such as drought conditions in Nevada, where that population of study in east of Reno is from. That in drought environments, you can have failure of reproduction that leads to a shift in the body size distribution that eventually has to play out and reestablish age structure as we go forward. So here's those same 17 years. I've repeated 2013 at the bottom and the top, so just so you can compare the arrows and see the shifts over time, because that's really what matters here is the average body size becomes the dependent variable. The changes in that average are really what is the trait of interest here. This is based on nearly 4,500 adult females, regardless of their tail regeneration, over those 17 study years. 2011 was an interesting year because of the large proportion of individuals that are small body size. And I should point out, this is all showing you proportion of body size in the y-axis, and these are all plotted on the same exact y-axis scale and the same exact x-axis scale for comparison. So what you see is the failure of reproduction of multiple clutches in 2011 meant that the recruitment into the population in 2012 was such that the average body size shifted by over 1 millimeter. And that is a relevant trait to see. What's happened is the age structure moved towards older females in the population. And that age structure stayed at that high body size for several years until such time as their age structure could be reestablished. Because what happened was the failure of recruitment in 2012 eventually led to a distribution like you see in 2015, where the population really is lacking a smaller age class. And by then, the older females have disappeared from the population, they've aged out of the population. So we get this really truncated distribution of body sizes that isn't on average different, although the shape of the distribution is different. That in 2015, we get a reproduction that can really benefit the population. Remember, 5 of the last 6 years have been years where they can get 6 clutches of eggs. And the years just prior to that, they were potentially getting that 6th clutch. There just weren't those adult females in that old age class who could have done so. So that what happens is 2016, when they get that 4th clutch of eggs, 2017 leads to a really left shifted distribution where we get another shift in the distribution downwards as we get the age structure reestablished in the population with those multiple peaks that are related to the ages. And then we get a stabilization of that population over time. So that's really the dependent variable then that we're trying to understand, changes in average body size over time. And there are a whole number of these variables that we can point out are potentially important for understanding that. The number of clutch iterations, either from the 1-year-olds the previous year, because they're numerically oftentimes the most abundant, or on a longer lag because of time lag effects. What I'll say is, I looked for time lag effects of up to 4 years, but really after 3 years, the explainability of those disappeared. So it was really a 3-year lag is what I mean by a longer lag, as I'll point out in a second. Or the older females, the oldest females in particular, either the previous year's clutch iterations, or again on a longer lag. The El Nino Southern Oscillation from the previous winter, or on a longer lag, the Pacific Decatur Oscillation from the previous winter, or on a longer lag. And then because there's potential for double whammies, the additive effects of those two, because we can get negative effects that sum to create changes in distribution, or get positive effects that sum. Then the last two are winter depth and length. And what I'll point out is these last two are the ones where I can start pointing out some of the limitations of my data collection, because winter length is the thing where it's not that it's not important, it's I don't have enough data to really answer the question. It's not that I'm not collecting data, it's I only have 7 years of data to point out how winter length has impacted these populations. And I will talk briefly as an aside how I'm doing that by using remote cameras and collecting data on the use of hibernacula, specific crevices that lizards come to consensus as the crevice to be in the fall. This time of year, lizards are slowly moving towards a certain crevice and coming to some consensus as to the crevice that is the warmest crevice for them to occupy. These aren't crevices that are used during the summer, they're only used during the winter. So they're specific dens like snakes use, but like you see here just above this bunch grass, a site where the animals come into the crevice. By the end of November, they're all in that crevice. There aren't really other crevices that are being used by the lizards. What this does, though, is it enables animals to be active all winter long. Side-boss lizards are supposed to be inactive for 150 days if you read the Pacific Northwest Herps by Brody, Nussbaum, and Storm. But January 27th, 2019, you see this picture where, if we zoom in, there are at least 12 animals that are coming out of that one specific crevice that is the crevice. You find these crevices by walking along the base of the cliffs at this time of the year, because you'll see them on warm days active in those crevices and maybe in a few other crevices. And then if you put a camera on them, what happens is they increase in number and then they slowly decrease in number as the winter gets worse and worse and worse. And then on certain days, on this particular day, it might have been sub-zero temperature. So my record is at the airport nearby, -5°C air temperature. 2 meters away from the cliff, -5°C air temperature. At the cliff, 8°C air temperature. Substrate temperatures in the 20s or 30s. The body temperatures of the animals are in the 20s and 30s. They're active because the microenvironment of the lava rock, the obsidian, warms up even in the sub-zero air temperatures because of the way the wind's blowing. And that's kind of what I'm trying to figure out, is what are the conditions that lead to activity. If you have cameras that are taking time-lapse photos, not the infrared activated, because they don't always activate the sensor, you have to use time-lapse photos and then record on the hour photos. This is the number of lizards per photo on the y-axis, days since October 1st. So showing you across the span of a winter, 3 different hibernacula. The green is an east-facing hibernacula. So what you can see is the animals increasing in number up to a point around early November, and then decreasing to a point. I plotted this so that 1 is the minimum, so that any dot on here is a photo that shows you animals that are active. So the animals disappear by about end of November from that particular crevice because it's east facing, and aren't active again until about day 130 or late February, potentially early March, depending on the year. In some years, there is no week of the year when activity doesn't occur, meaning inactivity is really relative. And in this particular year, 2018-19, that winter picture that I showed you in late January was here when these animals are coming out and being active. So what happens is a stereotypical pattern where they all converge on this crevice. They peak at some point in the fall. Each of these colors has a peak at different dates. They then enter what I call the valley of winter, where activity decreases, but never actually ceases for more than just a few weeks at a time, depending on local weather conditions, and then gradually increases again to a spring peak. So we have a peak to peak length of winter that, because I don't have enough data to demonstrate this, I'm going to exclude from further data analyses. But I want to point out is something that matters, because this is a behavior that is conserved evolutionarily, I believe, in the side-boss lizards. This past winter, I was in Big Bend, Texas. I was able to find animals that were active in early January as well. They'll come out of the ground burrows. They don't use rock burrows. They just use burrows in the ground in cut banks of dry washes, and will come out on days that are similar, 8°C where it's sunny. And they're oftentimes covered in mud because they're just crawling out of the earth. What's happening, I think, is an energetic conservation strategy, so that the animals will come out. When they're coming out, I've collected animals in Oregon in mid-January and immediately bled them to get their blood hormone levels, and being careful not to stress them out. The corticosterone levels in those animals is higher than anything you find during the summer. You can put them in a sock for 10 minutes and that stress level is lower than them coming out naturally first thing after a long period of inactivity. 3°C for a month is barely measurable corticosterone levels, meaning they don't have any cort when they're inactive at low temperatures. They come out, they get this huge cort spike because of the energy conservation strategy associated with cort. What happens is, I think, is a strategy where they're drawing down on glycogen stores and other carbohydrates to the point where they run out of those stores or come to a certain low, and they have to emerge on these warm days in the winter to scrounge those 3-carbon sugars or other byproducts, go through glycolysis and gluconeogenesis, which is one of the things that corticosterone does, and reform the glycogen stores that they then can mobilize at low temperatures. Previous studies that I've done in my lab show that they can last 150 days at 4°C without dying. So it's not the length of winter that matters necessarily, but at those temperatures, they don't mobilize lipid, they only mobilize the glycogen. And that's really what limits their length of inactivity in winter. So because winter is indeterminate length, and most of the animals that are coming out in the winter are young of the year, what seems to be happening is they're emerging so that they can regenerate those carbohydrate stores for that unknown length of future time that they're going to be forced to be inactive. And so winter length probably isn't the thing that matters for these animals. Winter cold could be, and when we do univariate correlations between the average body size and winter cold, just using a temperature in a deep crevice for every year that I have these data, it's not the previous winter that matters, it's a 2-year lag, potentially because of winter's cold on recruitment from the previous year and its impact on the age structure. But I'll point out in a second, winter cold doesn't really seem to be driving this either. Pacific Decadal oscillation does seem to matter, but on a 3-year lag. And so this is where I should point out what Pacific Decadal oscillation is. The Pacific Decadal oscillation is an oscillation, a fluctuation in temperature between a warm and a cool phase that you see there in the bottom in the temperate Pacific Ocean, and particularly over the Gulf of Alaska. Currently, this is today's picture. You see the blob out in the Pacific Ocean of warmer than average temperatures, but the current phase of the Pacific Decadal oscillation is slightly cooler than average. It's a long live phenomenon of typically about 15 years per phase, with about a 30-year oscillation from peak to peak in the period. So that this long live phenomena has the potential to drive this. A 3-year lag means that it's potentially what's causing the age structure in the population to fluctuate in those natural instances. But it could also be the El Nino Southern Oscillation. So with the same figure, we can see the same pattern because in this case, it's the tropical Pacific Ocean where we see these cooler than average temperatures currently because of the La Nina that's thought to be forming in the Pacific tropical ocean. Both of those impact populations. The El Nino Southern Oscillation has a much shorter period. Notice 2011, when we see that reduction in clutch number in the populations, it was really related to the El Nino Southern Oscillation being in this negative phase, but also the double whammy of the El Nino Southern Oscillation and the Pacific Decadal Oscillation both being in that negative phase. So that's why the additive effects has this potential, each of which has univariate components to it that I'll get to in a multivariate sense in a second. So I just want to point out what those are and why those might matter because of the short-lived and long-lived influences on the teleconnections with the weather inland in the Pacific Northwest. In terms of clutch iterations, is it the 1-year-olds that matter or the 3-year-olds that matter? The only thing that shows up on the univariate scale is the 3-year-olds, and it's on a 3-year time lag. That seems to be what's mostly driving the body size. When I take each of those that either are significant or approaching significance and put them into a multiple regression model to try to explain average body size, it's only the additive component of the El Nino PDO combined on a 3-year lag that really seems to matter. That is, the fluctuations that we're seeing are such that in 2011, when we get that great reduction in clutch number, that then plays out because it's disrupting the age structure of the population. That takes multiple years to reestablish, and it wasn't really reestablished until after 2015. That's when we get the population able to respond in an appropriate way to that changing environment. So if anything, this is pointing out the component of not necessarily climate change, but climate influence moving forward. If anything, this is the before time, that if we repeat this moving forward with the simple metric of average body size, hopefully we can see what's influencing these populations in terms of their life history. Not just life history. If I include now all of the unique individuals, the previous data was showing you some repeated individuals with palpations, but the unique individuals of over 7,000 animals that I've marked and recaptured over the course of the 17 years of the study, what I've done is not use market capture methods, but use, in essence, an equal effort survey of 19 to 21 days between mid-May and mid-July each year. And I'm simply showing you the total number of animals that I've marked each particular year. Recall 2011 being the worst year. What happened as a result of the 2011's failure of reproduction was that the population declined for another 4 years before it rapidly rebounded. And that's because the age structure had to reestablish before the population could take advantage of the permissibility of the climate to allow the fourth clutch to be produced. And what happened is that classic R-selected strategy where the population tripled in 3 years, from down around the low of 200 animals total that I marked in 2015, up to nearly 600 in 2017. And it stayed above 600 or near 600 for the last few years. So I've seen several oscillations of the population. So similarly, if we just use population size, what are the variables that affect population size? Go through that same process. The only univariate approaches of significance are not the 3-year-olds, but the 1-year-old previous year clutch iterations. Because numerically they're the most abundant in the population, that could be what's explaining that. And again, the precipitated oscillation, but not the El Nino, but again on a 3-year lag. When we take those two variables in a multiple regression model to explain population size, really it's only the PDO that even approaches significance. Whereas trying to explain the average body size was nearly 70% of the variation could be explained by that multiple regression. This is only about 30% of the variation, and neither of those things is significant. Because I only have 17 years worth of data, I think that's really the problem is each year is an N of 1 for this sample set. I need additional years to really get at additional oscillations because of that long lived nature of the precipitated oscillation to really tease apart the effects of these different climate modes. So that's the interesting thing is this isn't an endpoint for the study. This is simply a waypoint along the progression of how the study has gone from conceived to now trying to answer the question, how is climate change affecting individuals? By studying the energetics, by studying populations, and hopefully by studying assemblages of species, we can get at some of those questions. Life historical changes become very important then because we can try to understand how the timing and number of clutches and the size of those clutches, which I've studied in the past, impact populations. But it should be something that's simple and repeatable, because if it's a complicated metric of measurement, it's going to be really hard for someone in 100 years or 200 years to repeat. So simple body size on average as an illustration of information contained about population demography and what it tells us about the role of age structure by seeing how that fluctuates over time. Now that this study can demonstrate that, along with other studies that I've done, for instance, on populations in Nevada on drought, it's becoming clear that body size alone becomes a really important characteristic to understand that museum data play a role in understanding as well. So this all becomes part of the data legacy. Data legacy try to understand future change, meaning the future generations of biologists will only be able to answer that how question if today, here and now, we provide them with relevant data on those things that will be around in the after times, because these are the before times once the change has occurred. And it's an ongoing process, but in 200 years, in 300 years and beyond, hopefully the legacy of data such as these will be that studies like this are repeatable and the how question becomes more important and more relevant and more understandable. That only can occur if we understand what will and predict what will remain extant. And that's really the challenge is if I can give advice to a graduate student, it's make sure that what you're doing matters, that it's important, because otherwise, what's the point? So with that, I will end, and hopefully I have time for questions.
All right, thanks, Pete, for that stimulating talk. I was going to say we've already got hands up, but I'm going to use my prerogative as the host and I'll start one off. Let's see. Let me get up here. One quick one is, do the females ever become senescent? So if you got a 7-year-old female, she's still, do they ever become senescent, reproductively senescent?
To my knowledge, no. As far as I can tell, 7-year-olds are still reproductively competent. Whether or not they're producing fertile eggs, I don't know. But as far as I can tell, they're producing, for a 7-year-old, over 100 eggs over the course of their lifetime.
Yeah, pretty good output there.
Yes.
All right, then I've got a couple, and I might ask one at the end here, but how about this one? So you talked about court levels. I know you've been doing that with Susanna French. Mm-hmm. They're very low in the winter, but then the court level, or wait, I was going to say, is the court, maybe I missed this. The court levels are high in the lizards that come out, like on the rock there out of the, so that's to be able to, mobilize energy stores, I would think. Mm-hmm. So you were talking about, I forget what you talked about, but a lot of people talk about metabolic clearing or something like that, that they're somehow getting rid of metabolic waste. And what do you think about that? We have the same kind of thing with our garter snakes here, the local ones, like they'll come out in the middle of January. If you get 3 or 4, you're like, what the heck are you coming out for? You're just using up your stored fat. What's the point? So there must be some benefit to that. Any insights?
That's a possibility that I can't reject at this point, that there is some metabolic clearing going on, that the kidney function is temperature dependent as well. I do know that in vertebrates, all the way down to lamprey, amphibians, reptiles, the temperature dependence of some of those enzymes is such that it looks like only glycolysis can happen at low temperatures, and that they can't really utilize lipid stores. So I can't reject the hypothesis that it's not some metabolic clearing that's going on, but we don't see any animals that are clearing their gut, that are undergoing, that are pooping in essence. So I don't see that, which was what I would expect if that were true. And in fact, they can hold the pellets in their lower intestine, in the lab at least, for 70, 80, 90 days if you force them to. We've done some studies where we're giving them UV fluorescent powder to show that the last meal, they're not predicting when that they're going to end metabolic consumption of those things. What seems to be happening is they simply put it in their low gut where it's sterile and it can't really do anything to them. But that's certainly a hypothesis that at this point has potential.
Yeah, it's neat. All right, thanks, Pete. Let's see, I've got Virginia Weiss. I'm going to try to get Virginia to allow to talk. See if that works, Virginia.
Hello.
There she is. We can hear you.
Great. Pete, thanks so much for your really interesting seminar. I have two completely unrelated questions and big picture questions. One is, I'd be very curious to know the history, like in your thinking, the evolution in your thinking that eventually led you to the trail of thinking about these giant climate events. It's pretty, in the old days, it might have seemed really random, but I'd be curious to know how you got there.
That actually predates the study. So if you recall, the 97, 98 El Nino was one of the really big ones.
Got it. Okay. So
that's when I was a grad student at the University of Oregon, when, for example, Mapleton was cut off on 3 sides by the high river levels of the Sayusa.
Got it.
So that really got me interested in what is going on with this El Nino and how is that affecting populations of these organisms. So it was really, even predates the start of the study, my interest in how temperate environments are being impacted by something going on way down in the tropical Pacific.
Got it. Okay, my second question is related to your legacy comments, which I think are really very impactful. And that is, and I'm an experimental person. I don't study collections and I'm not super familiar with museums, but one of the challenges that I see that you might have going forward is, as species become threatened and perhaps endangered, how do you maintain the richness in the information in collections in organisms that can no longer be collected, or where collecting is way harder?
That's a really important question.
Are you going to change the type of information you're collecting? So I'd be curious to know what your thoughts about that are.
I mean, it certainly has, that problem has certainly altered my approach to the research that I'm doing. For instance, like toe clipping, which is a standard technique, is becoming harder and harder to justify to an IACUC. When I sat down in 1999, when I finished my PhD, and asked myself, well, what is it that is a model study organism in the lizards? What are the characteristics that they have? Widespread distribution, early maturation, small body size so that you can have them in the lab. But then one of the key characteristics was not endangered or threatened to become endangered anytime in the near future, meaning I can do the physiological studies that I knew I needed to do by consuming some animals in the process that wouldn't be possible on an endangered species. So that was something that really played into what is the key species to understand these long-term dynamics. It can't be something that's endangered, or else you're never going to be able to create a museum specimen or collect animals or do the physiological studies that I knew were relevant to create the understanding from molecular up to ecosystems that I'm trying to do.
Got it. Yeah. Thank you.
So careful consideration of species, I think, is the answer.
All right, I see Francis's hand up. Francis, I'm going to try to turn you on now too. Can you speak?
Hi, can you hear me?
Yeah, we can.
Awesome. Hey, Pete, I'm Francis Chan. I'm a marine ecosystem ecologist. Thanks for your talk. Really great to see the research just spanning the disciplines. My question is, the PDO, it's, we think of it as an ocean, of course, an ocean climate mode. But it's also one that we think of as integrating the high frequency atmospheric variability, because ocean has a memory. When you excite the ocean, it has a memory and is expressed through a very filtered low pass signal, such as PDO. But in your system, it's atmospheric processes. So it doesn't go through the ocean where you have that memory, the low pass filtering. Do you know, what's your guess on why why there is a memory? And I have a follow, maybe alternative answer.
So my understanding as to why the PDO has an impact inland, like on the terrestrial ecosystem, is what it's doing with the teleconnections is it's changing the low pressure system over the Gulf of Alaska in such a way that when it's in a certain phase, pushing the jet stream inland and changing the dynamics of how the rain is delivered. That seemed to be what's changing the fisheries in terms of the salmon return, because it's changing the flush out to the sea. And so it seems like what's happening is it's changing the position of the jet stream slightly inland and then out to the ocean in such a way that it's creating weather conditions that are permissible in the positive phase and hindering in the negative phase for the weather in the following spring to lead to an early onset of spring or delayed onset of spring. So that's my understanding of it.
Okay. So here's the what I kind of in my world we've come to understand that PDO it's a redshifted phenomenon. It's redshifted from almost a stochastic, if not the ENSO cycle, and we've had a hard time, in a sense, matching PDO to a lot of biological time series in a lot of ways. But it turns out we were playing the integration game wrong, because just as the PDO integrates the high frequency atmospheric variability, biology integrates the low frequency PDO variability, so that it's not just about phase shifting to ascertain the correlation between time series. It's actually another level of integration. So it's the integral of the PDO variability that it's a more important predictor. I don't know if you looked at that, but that might be a thing to follow, because the zooplankton people have been doing this with great success.
Yeah, I mean, that's a really important question. And I think that's a great idea. Because of the impact of these things on the age structure of the population, I mean, that's really what seems to be hindering the population from responding in appropriate ways. Like the climate might be permissible, but the population simply doesn't have the age structure that allows it to take advantage of it. So that seems to be what's creating noise in the system. And the fact that I found any predictability from PDO just on the mode itself, I think is speaking to the power of the long-term impacts of that on these inland terrestrial systems. But no, I haven't done what you asked in terms of looking at those integrals.
Yeah, it might be worth a look, but sure. Yeah, thank you.
I don't see another hand up, so I've got I've got one, and it actually follows along with Francis's as well. And so I have to admit, I'm still sort of running through these data in my head here. But from the long term, when you show the whole 17-year data set in that, I think it's in the number of encounters or whatever, how many you actually catch. You sort of started out low, like 2000, it started low, that went up, and then it dipped, like I said, in 2000, then it recovered. And now it looks like it's just so tantalized. It looks like it just peaked. And yeah, there you go. And now it looks like it's falling off. Of course, it's just really one year. And so it almost looks like, is it almost like you could predict this from your ENSO and the PDO? Or I was going to say, to throw a wrench in this, and everybody on this call knows that I'm not an ecologist, but could it also be just that in, in 2020, you're getting, the ecologists could do it, even the grad students, the first year grad students can do better than I can do on this. Is it like, is it a competition thing, or what, it's just like, it's a density, what would it be? Density dependent kind of interaction you're looking at there. All right, so I think there's a question in there somewhere. So it's kind of, is it predictive with the, those big cycles? But the other one is just the null hypothesis. Is this just looking at, I shouldn't say just looking at, do you think something like competition or whatever is playing some role in there? Anyway, I'll shut up and let you talk.
So the short answer is I've been trying with limited success to predict the next year population for years, because I have to tell ODFW how many lizards I anticipate collecting. And I'm regularly underestimating that, so that I'm always collecting more and then saying, I'm sorry, I collected more than I thought I would. So I've had very little success at trying to do that, to try to predict how many animals I'll collect the next year. There's definitely a density dependent mechanism, because what I didn't point out was I've also been sampling the north facing slope of Wright's Point, that study site. And what I find is in years like 2010, 11, there were almost no animals on the north side. So there's almost nobody who can survive on the north side in those years. And then in recent years, not only are more animals showing up from year to year, so clearly crossing that 200-meter wide lava flow and establishing on the north side, they're also establishing in the middle, in the sagebrush flat in the middle there. And year to year, I'll find the same animals on the middle as well as the north side, to the point where I get recaptures of animals that are established on the north side that seem to be potentially creating a novel population on the north side that is driven in part by the recruits that are simply flowing out of the high density population to the south. So the north side is a valve. The pattern looks almost identical to this, meaning we see the same oscillation. It's just, it goes to 0 in those bad years, and then it pops back up to non-zero numbers, as many as 50 along the north side in those years when it's in, 600. So there's like maybe 10% of the population that's also establishing on the north side in years where it's above about 200 or 300.
Very cool. Yeah, that's neat.
So certainly density-dependent mechanisms going on in the population that could be regulating it and could be explaining why we get a decrease in that most recently.
Right. I was going to say, so just to having been someone when Pete talks about going out there in the desert, and a few of our grad students and labs here work in Eastern Oregon, so they know what it's like to work out there. Having gone to grad school in Texas and been out in beautiful Santa Elena Canyon there, I recognize that immediately. It's hard as heck chasing these darn lizards out in the desert there. It's like it's enough to even as young people, I remember just being busted down. I never worked so hard in my life chasing lizards and things. So I do want to say that for all the grad students in the.
So can I just interrupt and say, here, in Oregon, I can find 30 animals per kilometer when I hike out in Eastern Oregon. In Texas, in the winter, I was getting 1 lizard every 2 kilometers. Yeah. Meaning I would get maybe 2 lizards a day in the winter.
And it gets cold out there in the winter. Mm-hmm. All right. So we're kind of at the top of our hour here. So again, if people have some burning desire to get something, just write to Pete. I didn't put his email. I should have put his email in the chat here, but anyway, you can just catch up with me, and you folks are all smart enough to figure out how to get a hold of Pete. But if you can't find him, I can get you to that. And so, Pete, I just want to thank you for that great talk, and for people that ever want to really meet him in the present or in the flesh, I should say. Pete comes through every year because he's making the pilgrimage out to Eastern Oregon. So I look forward to seeing Pete. So, anyway, thanks, Pete, very much.
It was fun. Take care.
All right, we give a digital and a vocal applause here. All right, thanks everybody for coming. All right, I guess that's it. So Pete, I'm going to end this all right in here. We'll catch you on the flip side.
Take care. Have a good winter.
All right, you too. All right, bye now.